In order for drugs to get from where we place them in the tissues, either parenterally or um, having them breathe it in or uh, given orally, there are four different types of transport that are used to allow drugs to move around in the body. Here are their names: passive diffusion, facilitated diffusion, active transport, and physical transport. So, as the name implies, in passive diffusion, in my little kitty picture in the previous slide, um, passive is a way that we're very familiar with this. So if you have coffee in the morning and you put cream in it, when you put that cream in, it just starts to spread throughout the coffee. Sometimes we stir it in, but it typically spreads on its own, and the coffee goes from being a sort of clear, dark color to an opaque, milky brown color. So that is just simply that the molecules have of the milk of the cream have mixed right in with the coffee and have spread until they're of equal concentrations. So that's what happens with passive diffusion. But we're talking usually about moving across a membrane, and there is no energy that's used to do this. It just moves depending on the concentration gradient. That's what drives it. Um, This is an illustration of that. So this is a bilipid uh, cell membrane, and um, there is uh, these molecules that you'll see go and they can't make it through. It's simply because they can't go through that membrane. So there's one that goes through the membrane. What property does it have that allows it to go through the membrane? Well, the membrane being bilipid, it um, likes the same thing that it is. So it has to be lipid loving or a lipophilic. That one was just one that passed through. That's a lipophilic molecule. So the lipid nature of the membrane does not allow something that is water loving, or that's called hydrophilic, to pass through. But if it's lipophilic, it can pass through. So this red piece may not be allowed through. That was um, an example of our next thing, that little red thing that went through the membrane protein. But the passive diffusion, that is a passive diffusion right there. It just goes right through the membrane, and at the end of a molecule, a group of molecules going from this side, to outside of the cell to inside of the cell, the concentration will be equal. This diagram, or this animation, implies that it's just going one way. In passive diffusion, it goes both ways, and basically until the concentrations are equal on both sides. So even though it passes in both directions, the net effect is that the same concentration on both sides. All right, so to facilitated diffusion. Um, in this one, it is still diffusion. There is no energy expended, just like in passive diffusion. So no energy is expended. The thing that drives it is something called a carrier molecule. And if you remember that red um, six-sided figure was moving through, and it was moving through with a carrier molecule. So in this case, again, it's got to be lipophilic, or have this affinity. I shouldn't say it's lipophilic. It has an affinity to this carrier protein. And the end effect is, let me see if I can get it to go again. I'll have to go back, and then it doesn't seem to keep going like the previous one. These are being moved across until we have equal concentrations on both sides. So it's again driven by concentration and this carrier molecule. There is no energy expended. It's just that that carrier molecule has an affinity for the other molecules that are being taken across, And then once they reach the same concentration on both sides, that is when it stops. There may be some carrier molecules carrying it to the other side, but the net effect is it's an equal concentration on both sides. So that's what drives it, is that concentration gradient. Again, just like passive diffusion. So active transport, as the name implies here, we are going to expend some energy. The two previous types are the most common types. So facilitated diffusion and passive diffusion are the most common ways that drugs move through membranes in the body. Active transport, though, does have, we do have some drugs that are carried this way, and it actually allows them to accumulate in certain areas in the body, that is more than the concentration gradient. It pulls it into a particular tissue. That can be useful, but it also can create some toxic situations, that we'll be talking about when we talk about certain drugs that have an affinity to collect in different areas of the body. So this is an illustration of that, and you can see energy is needed. It's similar to facilitated diffusion, but in the end, more of the drug ends up inside of the cell than outside of the cell, because it is not driven by the concentration. It's driven by the affinity of this molecule and the energy that pulls it to the inside of the cell. So this is just kind of another little illustration of, these are two passive types of transports. They're driven by the concentration, and as long as there's a high concentration on this side and a low concentration on this side, the drug will be pulled over to the inside of the cell. And when they are equal on both sides, we might have movement back and forth, but the net effect is that both sides will have equal concentrations. In facilitated diffusion, here's our carrier molecule. Same thing, driven by the concentration until we get equal amounts on both sides. And then active transport, you can see that most of the drug would be pulled into this type of tissue, and not driven by the concentration gradient, but just by energy being used to pull it across the membrane. All right, and lastly, and this is an uncommon type of transport, but it does exist, is pinocytosis, which is called cell drinking, or phagocytosis, which is cell eating. In this, energy is used to bring molecules across membranes, because it takes a lot of effort to do either one of these. Pinocytosis is the way that we think vitamin B12 is absorbed, from the gastrointestinal tract into the body. And phagocytosis is used for bringing large proteins, and typically we're talking about antibodies. One area that we think this occurs is young animals getting antibodies from the milk of the mother. And that's how they're relatively large molecules, and they're being brought across the GI cell membrane by phagocytosis. So let me show you how that works. So this is a membrane that just basically engulfs a group of molecules and then breaks off, and we have a little vesicle that is brought inside of the cell, but we've essentially drunk some molecules into the cell. And phagocytosis, you can see that this is much larger than these other smaller molecules. Something like an antibody is basically engulfed and then taken into the cell. So not super common, but it does exist as a way to get in there. I'm going to have you fill out this chart.